Hello and welcome to this webcast, which is part of our series on oral availability of medication. I'm very pleased to be joined by Dr. Urvi Gala today. She's a principal researcher at Disposol Technologies based in Austin, Texas. Dr. Gala, many thanks for taking the time to speak to us today. Thank you.
Thank you, Alistair, for having me here. And I think LeadWire is a great platform, so I'm happy to be here and share my research.
Thank you. And your research focuses on improving the solubility of various anti-cancer drugs, such as abiraterone. Can you first tell us about the background and key findings of your recent research?
Sure. So let me give you all a bit of a background and then I'll get into the key findings. So the drug abiraterone is an active metabolite of a prodrug abiraterone acetate, and it's used in treatment of prostate cancer. Its commercial formulation is Zytiga, which has abiraterone acetate in crystalline form, and it has an estimated oral bioavailability of less than 10%. Now, it has been shown that when we increase abiraterone exposure, the therapeutic outcomes are improved. And however, when Zytiga's dose was doubled from like 1000 milligrams to 2000 milligrams, the increase in drug exposure was merely 8%. So the complete therapeutic potential that abiraterone has not been realized, and that's due to its formulation limitation. So the goal of our research was to develop an abiraterone formulation that has improved dissolution as well as improved oral bioavailability. In order to achieve this goal, we used kinetosol technology. Kinetosol technology is a solvent-free process and it's thermokinetic, and we use it to develop solid dispersions of drugs with different polymers. And in this case, we used conventional polymers as well as oligomers such as cyclodextrin. And what we found was that while most of abiraterone kinetosol process solid dispersions improved its dissolution, but the oligomer-based solid dispersion improved the dissolution by 15 folds as compared to the crystalline drug. And then when we performed in vivo studies in dogs, we saw a bioavailability enhancement of 4x, that is like 4 times as compared to the commercial formulation. So basically, we were able to develop an abiraterone formulation that had improved dissolution as well as bioavailability.
Why is it that drugs that have high efficacy are often poorly soluble? And do you think it would be maybe better to prefer drugs with lower efficacies but higher solubility over drugs with higher efficacy that are poorly soluble?
So yeah, firstly, that's a great question. And I'll answer this question in 2 parts. So first, let's take your first question, which is that why drugs with higher efficacy tend to have poor water solubility. So there are several reasons for this, but I'll focus on 3 main reasons. So the first reason is for a drug to exert its effect, it needs to reach its target site of action. And in order to do so, it needs to cross cell membranes. Now, to cross cell membranes, the drug does need a certain degree of hydrophobicity or lipophilicity. And generally, when the drugs that have higher lipophilicity will tend to have poor water solubility. So that's one thing. Second thing is, for several drugs, hydrophobic groups are essential for them to exert their effect. For example, let's take anti-cancer drugs. So several anti-cancer drugs have very bulky and very complex hydrophobic groups, such as polycyclics. And those are essential for the drug to actually bind to its target receptor. And as a result, these hydrophobic groups impart poor water solubility to the drug. So these are like 2 reasons related to the chemical structure of the drug. The third reason is related to its physical form. So several drugs are supplied in a crystalline form. And the reason why people do so is because crystalline form is more stable and has higher purity. Now, in order to dissolve this crystalline form, high energy is required. And as a result, the drug has poor dissolution. So these are like the three main reasons why drugs which have higher efficacy tend to have lower water solubility. Now let's get into your second question, which was that whether we should give higher preference to efficacy or water solubility. So, in my opinion, we should give higher preference to efficacy. That's because it's like a key essential to developing good therapeutics. And then we can address the problems that arise due to poor water solubility by developing better formulations. So, efficacy is definitely the prime goal.
What are some of the limitations that arise due to the insolubility of drugs? Does this arise due to oral administration, and can such limitations be avoided by using other routes of drug administration?
Let me address this question by sharing a slide. So, firstly, let's talk about the limitations that arise. The first limitation that arises is poor oral bioavailability. So, here I have a graph of plasma concentration versus time. Now, when you give a poorly water soluble drug directly intravenously, the amount of drug exposure is going to be equivalent to the area under this blue curve. Now, when you give the same amount of poorly water soluble drug orally, it's going to have significantly less exposure, equivalent to the area under this red dissolution curve. So basically, the availability of the drug is going to be less, which is the poor oral bioavailability. Now, moving on to the second limitation, which is pharmacokinetic variability. What it means is that due to poor water solubility of the drug tends to have very erratic dissolution. As a result, the plasma concentration values vary a lot within same patient as well as between different patients. So that's like a major limitation. The next one is nonlinear pharmacokinetics. What it means is here's a graph of drug exposure versus drug dose. In an ideal case, we would expect that the drug exposure increases linearly with a drug dose. But when you have poorly water soluble drug, which is presented in a conventional formulation, what happens is the drug exposure does not increase linearly with drug dose. And in fact, after a certain dose, the exposure plateaus. So you cannot actually have enough exposure. Then the next limitation is efficacy. Now, for several drugs, the efficacy is directly proportional to the plasma concentrations or the drug exposure. And due to the poor oral bioavailability, those plasma concentrations or drug exposures are not reached, and hence you will have poor efficacy. And then the last, but certainly not the least, is toxicity. So by toxicity, what I mean is that when these drugs, due to its erratic dissolution, have variable plasma concentration, and as a result, several times the concentration would cross the minimum toxic concentration level. As a result, the patient could have side effects. So these are like the 5 major limitations that occur when a poorly water soluble drug is delivered orally. Now, to answer your second question, that can these limitations be avoided if we, use another route of delivery? So the answer to that is yes. Maybe we can use, like, let's say, intravenous drug delivery and avoid these limitations. But then we need to note that these alternative routes have their own limitations. And generally speaking, oral drug delivery is the most preferred route of drug delivery method.
Your research focuses on amorphous solid dispersions. Can you tell us how exactly this works?
So first, let's talk about the general strategies or formulation techniques that are used to address poor water solubility of drugs. So these include particle size reduction, developing a salt form of the drug, then developing co-crystals, or adding solubility enhancers like surfactants or co-solvents, also developing lipid systems, and then, of course, developing amorphous solid dispersions. Now, amorphous solid dispersions are my key area of research. And before we get into how exactly the amorphous solid dispersions work, let's understand what it means. So, amorphous solid dispersions are basically a system wherein the API, which is the active pharmaceutical ingredient, or the drug, is molecularly dispersed in a polymer. This slide basically depicts the general process of drug solubilization, which involves four major steps. The first step is structure disruption, which is the breakdown of the structure, followed by solvent cavitation, which means that spaces are made in the solvent for the drug to dissolve. Then the next step is solvation, which is actual process of drug solubilization. And then the last step is maintenance of that solvated state. Now, when the drug is presented in a crystalline form, high energy is required to disrupt or break down this whole crystal lattice in order to get amorphous or non-crystalline form of drug. And even when we get this form, this is highly unstable and it tends to go back to its crystalline form. And I would just use my laser pointer so that it makes more sense. So when this non-crystalline form undergoes solubilization, any amount of drug that is higher than equilibrium solubility then tends to precipitate out. As a result, the total amount of drug dissolved is very low. So one would say, how about, we give the drug in direct amorphous form? So when we give the drug in direct amorphous form, it definitely avoids this major energy requirement step of structure disruption. But what happens is this amorphous form is also very unstable and the drug tends to recrystallize back into its original crystalline form. Also, when this amorphous form solubilizes, it gives us supersaturated concentrations, but those are very momentary. And eventually, the drug precipitates out. Now, in the third case, when the drug is presented in form of an amorphous solid dispersion, what happens is the amorphous form does not recrystallize back because the polymer present in the solid dispersion prevents drug recrystallization. Also, it avoids this major energy requirement step of structure disruption. Also, the solvent cavitation step is improved because higher degree of cavitation occurs. And eventually, we get supersaturated drug concentrations that are dissolved. And the drug precipitation is also avoided because of the polymers in the system. So this is how the amorphous solid dispersions work.
Dr. Gala, many thanks. Let me wish you and the team at Dispersol Technologies all the very best. Thank you.
Thank you.